In this video we're going to look at sequence degeneracy. That is, what happens when you back translate a protein sequence to a DNA sequence. Degeneracy is just a reflection of the number of different molecules of DNA that could encode that one single peptide sequence. As you know, we have our amino acids, and each amino acid has one or more codons. These codons are triplets of bases in DNA that encode for that particular amino acid. So let's say we take alanine. Alanine has 4 codons: GCT, GCC, GCA and GCG. Here we've got one amino acid and it can give 4 different DNA molecules. So here we would say the degeneracy is 4. Now let's take a very small peptide consisting of only 2 amino acids, alanine and cysteine. Alanine, as we've already seen, consists of 4 codons. However, cysteine has 2, TGT and TGC. Now if we take the 4 codons from alanine, each one of those could pair up with a codon from cysteine. So this would effectively give us 8 DNA molecules. So in this case, one peptide has given us 8 different DNA molecules that could encode for that peptide. Basically, it is 4 times 2, which is equal to 8. That is the number of codons of an amino acid, in this case alanine 4, multiplied by the number of codons of the next amino acid, cysteine, which is 2, which is equal to 8. Now it is possible to express this degeneracy where we've gone from one peptide to 8 DNA molecules as a single strand of letters. To do this, we have to use the IUB code, which is shown here. If we look at the 8 molecules of DNA that's required, if we write out the code, then what we get is GCN, because in that third position there can be any base, TGY, because in that last position there could only be a T or a C, and this matches what is seen in the table. So that's it. That is how you calculate the degeneracy of a translation from a protein sequence to a DNA sequence, and I've also shown you how you could express all those molecules that could encode for that protein as a single line of letters.